Okay, so now we understand the basics in translation. Here's the steps. If we want to make a protein, this is what we have to do. Okay, first we need the participants. We need some messenger RNA. That's the transcript. That's the information that codes for the amino acids we want to make. We need some transfer RNAs. Remember, transfer RNA is what carries in the amino acids, brings them into the play. We need some ribosomes. Ribosomes are where protein synthesis occurs. We need some energy, and that's going to come in the form of ATP. And we need protein factors. Okay? All of these are going to assemble during what's called translation initiation. They all sort of gather at the transcript and form a translation complex, which is then going to begin the work. So the first thing we have is we have messenger RNA, and that's the transcript, and it hydrogen bonds to a short sequence of ribosomal RNA in the small subunit of the ribosome. So remember, a ribosome is made of two subunits. It's made out of RNA, which means it has exposed bases. Well, there's a section of the messenger RNA transcript that's added that attracts a certain section of RNA bases in the ribosome, the small subunit of the ribosome, and they hydrogen bond together, kind of hold each other in place. The first codon in messenger RNA that's actually translated is AUG. So there's always an AUG at the beginning of where we want to start making our protein. AUG codes for start. Now, AUG also codes for methionine. So often at the beginning of a protein is the amino acid methionine. Not always, but a lot of times it's the first one there because it's also coded for by AUG. So when there's an AUG in your messenger RNA, a transfer RNA with the appropriate anticodon, which would be what? What's the anticodon for AUG? Right? Once that comes in, it brings in methionine with it. And so oftentimes we have the first amino acid is methionine. So we have this messenger RNA piece, we have the ribosomal RNA, the small subunit, and the transfer RNA with methionine. That's called the initiation complex, and that attracts the larger subunit of the ribosome. So the large subunit comes in, it attaches to the initiation complex, and it begins to sort of ratchet its way down the messenger RNA, bringing in tRNAs with their amino acids, matching them up, and creating essentially a long chain of amino acids. So the tRNAs match up their codons with their anticodons with the codons on messenger RNA. They're each attached to an amino acid, and we get this long growing chain of amino acids. There are enzymes that come in and link them together to form peptide bonds, and we get this long chain of amino acids being formed. That's called elongation. Finally, when we reach one of the stop codons, UGA, UAG, or UAA, elongation stops. There's no anticodon, no tRNA molecules that have an anticodon for the stop codons. So there's nothing comes in at that point. And when there's nothing there, the ribosome just sort of detaches. And now you have the transcript, which could be used again by another ribosome. And you also have a new polypeptide string of amino acids. Now, it's not a protein yet, but it's well on its way.
Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific three-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin,